Hi, welcome to your lecture on DNA replication. This is the process of taking one molecule of DNA and making two. You know that your DNA needs to make a copy of itself so that each individual cell can have a full copy. And now we're going to find out exactly how it does that. DNA replication is the process of copying a DNA molecule so genetic information can be passed on from cell to cell. DNA replicates before the cell divides during the S phase of interphase. If you remember, interphase happens before mitosis and meiosis, or actually in between rounds of cell division, in between rounds of mitosis and meiosis. And it is divided into three phases: the G1, the S, and the G2. S stands for synthesis, and that is when a new copy of DNA is made. When you synthesize something, you make something, and that is a copy of DNA is being made during the S phase. So, quick review of our last lecture. DNA is made up of two strands of nucleotides, and they are complementary strands, with A always pairing to T and C always pairing to G. And they're held together with hydrogen bonds between each of the bases. So how does DNA replicate itself? Well, each strand is used as a pattern or a template to make two new DNA molecules, and this is called semi-conservative replication. It is semi-conservative because you are using one of the old strands and using it as a template for creating an original strand. So here you have a picture of the original left strand with a new right strand, and then the original right strand with a new left strand. The first step in DNA replication is that the double helix will unwind, and then an enzyme called DNA helicase will unzip the DNA molecule, basically going between all the hydrogen bonds between the bases and breaking all the bonds, so it becomes two single strands. The yellow guy right there is the DNA helicase enzyme, and then there's a picture of the unpaired bases. The next step is that another enzyme called DNA polymerase will take free-floating nucleotides that are in the nucleus, and remember, a nucleotide is a sugar and a phosphate and a base that are attached to each other, and that base would be either adenine, thymine, guanine, or cytosine, and it's going to pair it up with the now free strand. So it'll take the free nucleotides in the nucleus and pair them with the nucleotides on the DNA to form new hydrogen bonds. The nucleotides that are in the nucleus free floating come from all of the food that you eat. So you are what you eat. So the steps in DNA replication, here's another picture of it. Here's your original strand. It first unzips using the enzyme DNA helicase, and then DNA polymerase, a new enzyme, will come around behind it and pair up the free-floating nucleotides. And this animation is showing kind of how the nucleotides pair up. And that will happen on both sides of the DNA molecule. And so you start with one original, and then you have two identical copies. Last but not least, the DNA will wind back up into a double helix, and the final products of DNA replication are two molecules of DNA, each having half new material and half old material, which is why we call it semi-conservative. This is another picture of DNA replication. You can see the enzyme DNA polymerase riding along the DNA molecule. It goes in two different directions depending on which strand it is on, and it's pairing up free-floating nucleotides. Once you have two copies of the DNA, all the genetic information can be passed on to new cells during cell division. So once that happens in the S phase of interphase, the cell will go through either mitosis or meiosis. If it is a body cell, it is going to go through mitosis. If it is a germ cell, and the purpose is to create your gametes or your sperm or your egg, then it will go through meiosis. Polymerase chain reaction is a type of DNA technology that can be used during forensics and paternity testing to replicate millions of copies of DNA from a very small sequence or sample. This link that says PCR video, if you download my PowerPoint slide and click on the link, it will show you how the process of polymerase chain reaction works and how you can take a little sample of DNA that you get at say a crime scene or for paternity testing and create a whole bunch of that strand so you can do a lot of different testing on it. So the DNA is being replicated all the time in many different cells in our body, and there are lots and lots and lots of pairs. So the odds of having a typo are actually very, very small. Replication is extremely accurate. Only 1 in a billion base pairs are incorrectly paired. And when the mistake is made, the DNA molecules can correct itself, so the genetic codes remain the same. There are enzymes that proofread, and if you have a wrong pairing, say an A bonds to a G, and that is incorrect pairing, then you have enzymes that can go through and cut out the wrong base and substitute and put in another one. But it does not fix all of the mistakes, and in a different lecture, we are going to talk about all of the different mutations that can occur when the mistakes are not caught. Okay, have a great day.